So this audio lecture is meant to be kind of a crash course in microbiology. So if it's been a while since you took your Intro Micro 304, it's just meant to kind of fill in any of those rusty, cobwebby areas. And just focusing on the microbes and the structures that we'll talk about during the course of the semester as how it applies to food microbiology. So there are four basic types of microbes that are found in food. There's bacteria, yeast, and molds. Both of those are fungi, and viruses. Okay. Now there are some algae, protozoans, and also hemolyphs, or parasitic worms, nematodes, that are also important in food, and we'll talk about them later on in the semester, but the focus for this audio lecture will be about bacteria, fungi, and viruses. Algae are important because there's some that actually cause shellfish poisoning and fish poisoning, so it becomes important when we're looking at contaminants of those food sources and foodborne illnesses. Also, there's several forms of algae that are utilized in bioprocessing because they're sources of vitamins and also protein. So there's algae that are good sources of single-celled protein. So we'll talk about algae later on when we talk about fermented foods and food microbes. So we want to keep in mind with microbes, they come in different sizes. So this figure just shows you if we were to use the light microscope, which we traditionally would use in our micro labs, you can see that we're able to see bacteria, but that's in the kind of the end of the scale of where the light microscope can be used. So light microscopes, when we're looking at eukaryotic animal cells, plant cells, is going to be much better at being able to see that. So for bacteria and viruses, we'd have to go up to the high power, and even viruses, we're not going to see that well, and we'd also would have to culture viruses with a host cell. So for many reasons, we wouldn't look at viruses in our lab. In order to see viruses and then smaller structures or proteins, we would need to use an electron microscope. So just keep in mind that the size is going to vary on these organisms, whether they're prokaryotic, which are bacteria, or whether they're eukaryotic, which is our yeast, molds, and viruses. So again, when we look at microbes in the food, it's going to be the bacteria, yeast, molds, and viruses. And it's because of their ability to cause foodborne disease, food spoilage, or actually produce food or food ingredients that they become important when we look at food. And bacteria are going to be the largest group just because they're ubiquitous, which means they're present everywhere. And also they have a rapid growth rate. There are also puron or protein-based infectious agents. So those become relevant, and we'll talk about those when we talk about foodborne illnesses later on. So you've probably heard of like mad cow disease. There's also chronic wasting disease that's been seen in elk that can be transmitted to humans and other animals. So it's also relevant. So if we look at the phylogenic tree for microorganisms, they're going to be found in the three major divisions. So as you can see, there are three divisions here. We have bacteria that are going to be present, and so we already mentioned that bacteria are a large group of them. You'll notice that there's some different names in them. Some of these we'll talk about later on in the semester and throughout the semester, like Clostridium. But there's also bacilli and actobacteria. There's also the archaea, and so you can see from their names, there's methylobacteria, halobacteria, and you also have this thermal. So like these are really exotic prokaryotes. They like extreme environments, so they don't tend to play a role in food microbiology, and even in a medical microbiology class. So it's really in an environmental microbiology class that you would see archaea being more pronounced. But again, there's definitely microorganisms that are present there. And then you have the eukarya. Eukarya would contain our fungi, but also those protozoans. And we're also in there, but again, we're not talking about microorganisms, not humans. When we look at microorganisms, the nomenclature, how they're named, is going to be in two parts. So this is known as binomial nomenclature. So the first part is going to be the genus, and this is written with a capital letter. And then the second part is the species, and this kind of becomes an adjective used to describe, provide more information about the genus. Both parts usually have kind of a Latin base to it. And when written or typed, they should be italicized or underlined. And you'll notice that sometimes, even I forget to do that. So just do your best with that, especially, sometimes in some of the settings for Canvas, you can't actually change font size and italicize things. So don't worry tremendously about that, but just be aware that normally you should italicize or underline it. So examples you can see here, we have penicillium, and so penicillium is our genus. So there's lots of different types of penicillin, but then we have specific genuses for that. Lactobacillus acidophilus, that's a probiotic. That's pretty common. And so again, Lactobacillus is the genus, Acidophilus is the species. Acidophilus tells me that, with this bacteria, there's something about acid. So Phyllis likes, so it likes acidic environments. Lactobacillus, that genus tells me that it's bacillus rod-shaped, and we'll talk about that in a moment. But lactic acid, lactulose, and so this would be a bacteria that's likes acidic environments or creates an acid, and it's found in like milk, dairy. And if you looked at it in a microscope, it would look like a bacillus. It would look like a rod. Okay. With bacteria species especially, they can be divided into subspecies. So sometimes you might see this sub-SP or SSP, and again, that just provides you with more information. So there's different subspecies of Lactococcus lactis. And so again, just like, we have humans, and there's little variations between us. So bacteria can have enough genetic differences or characteristic differences that are unique, not just when we look at the genus level, but when we look at species level to the subspecies level. And sometimes we even have strains that are unique variations among that. So we're going to look first at fungi, so looking at morphology and structure of fungi. So fungi are going to be eukaryotic, so they're very much like you as human beings. With yeast, these are going to be unicellular, and molds are going to be multicellular. Okay, so both of these, yeast and molds, are examples of fungi. And so later on in the semester, when we're talking about controlling growth and making sure there's not contaminants present, we can have antifungals, and they could work against yeast and molds, but then we might have a product that would only work against the yeast or only work against molds. So we'd have to keep in mind what is present that we need to fight. Now, fungal cells have cell walls. Those are quite rigid, but then they have thin plasma membranes, and so that cell wall really provides its structure. The cell wall is going to be composed of carbohydrates, where the plasma membrane is going to have sterols present in it. So it does have the phospholipids, but there's also sterol present in it, which helps to stabilize it. The cytoplasm is mobile, so there's streaming, so it does have a developed cytoskeleton that allows for the movement of vesicles through it. so transport is present. It also has organelles as a eukaryotic organism, so it has mitochondria and also vacuoles, and those have membranes on them. When we look at the ribosome of a fungi is going to be 80S. So the two subunits, the large subunit would be 60S, and then the smaller subunit would be 40S. But when they bind together, their overall size is 80S. Again, that's something that we would see in eukaryotic cells. And those ribosomes will attach to the endoplasmic reticulum. And again, if we think about comparing this to bacteria, this isn't the same bacteria. With its DNA, it's linear chromosomes, and there's histones. So the chromosome, the DNA is wrapped around the histones to package that, and that's going to be enclosed in the nucleus through the nuclear membrane that has nuclear pores. And then with fungi, just like eukaryotes, their cell division can be by mitosis, and that would be asexual reproduction. And then for sexual reproduction, when it occurs, because it doesn't always occur, it would be through meiosis. So looking, those are shared characteristics between the fungi. So now looking at molds. So molds, again, these are the multicellular organisms that fall into the fungi category. So these are going to be non-motile. They're usually filamentous, and they can actually be branched. So you can see that in that figure below. This is an example of a mold, penicillium, and you can see that kind of stalk. So this is going to be multicellular, and then there's branching out. And then right at the end is where we actually have spore productions forming. And so the shape and size and also the color of the spores are actually used for classification. Okay. For molds, their cell wall is going to be composed of cellulose, chitin, or both of those. Okay. So again, what's present in the cell wall can help identify. Off of the main thallus of the mold, you can get a large number of filaments. These filaments are known as hypha, and this is what gives a mold kind of their velvety, fluffy appearance. So if you've ever seen mold growing on like oranges, which is actually in the first slide, the opening slide, or if you've had, you left something in the back of the fridge and seen there's mold growing, it's going to have that velvety appearance, and it's that hypha that kind of gives it its height. And some of those hypha are reproductive, and so it's actually going to extend out, and so again, that's what you're seeing here. And then they can form exospores, and those spores then can be released. And again, the spores can be used for identification because the shape, size, and color of them are unique to each species of molds. Now looking at yeast, like bacteria, these are widely distributed in nature. So if you took a sample from virtually anywhere, yourself, food, soil, in your house, you'd probably find yeast present. So yeast, again, are single cellular, so unicellular organisms. And so they're going to have anywhere from an oval to spherical to elongated shape. And they're going to be they're eukaryotic, so they're relatively large in size, and so if you were to look at them underneath a microscope, you would be able to see them at 40x. So usually if a student in a microbiology class is like, they've got their microscope on 40x and they're like, I can see them, I can see the bacteria, it's like, it's probably not bacteria, it's probably yeast, because again, the eukaryotic organisms are going to be larger. These are non-motile. And their cell wall is going to be made up of polysaccharides or glycans, proteins, and lipids. So the composition is a little different than what we saw with our molds. The cell walls can actually have scars on them, which will indicate where there's sites of budding. And so the budding is where we have asexual reproduction occurring. So the cell is going to basically divide, but they don't evenly split the cytoplasm and material, so the new cell gets like a smaller amount, so it's like a little bud. And if you look down in the image here, kind of a little bit to the left of center, you can see how there's that smaller cell coming off, and that would be one that's budding. And so what actually happens is you get that scar. And so if you, especially if you look under like scanning the electron microscope, if you look at the surface of that cell wall, you can sometimes see how many rounds of cell division a yeast has undergone, how many budding events they've carried out. And there are different chemicals that you can use to prevent budding and also promote budding. So if you're looking to augment yeast in its growth cycle, you can do that. So now looking at bacteria, and we'll spend a little bit more time on bacteria because it's such a large group and such a focus of food microbiology. So bacteria are unicellular. They're known as prokaryotic. So these are quote-unquote simpler organisms because they lack organelles. They have three main morphology forms. So they have cocci, which are spears, rod shapes, which are bacillus, and then they can have this curved comma-shaped, which is vibrio. So if you look down the figure below, you can see those three. There are some additional variations of those. So I said three because that's what the textbook, the food microbiology textbook says, but I can't lie. So there's also cocci bacillus, and so these are kind of elongated. They're not quite bacillus, but they're not quite spears, so this kind of intermediate. There's also spirillum. So this is where we kind of have a comma, but there's another little kink in another comma, and so it has more of a kind of wiggly type appearance to it. And then we have the spirochetes. Now the spirochetes are very interesting because they actually have these flagella that are embedded into them. And so when they wiggle, they actually move in a corkscrew motion. So you can kind of see that corkscrew appearance on that figure. So they're really important in medical microbiology because spirochetes tend to be, because they can corkscrew, they can get into different tissues, and so some aggressive diseases like syphilis, like leptospirilla, like Lyme's disease are caused by spirochetes, and it's because they can bore into these tissues and then they can evade the effects of antibiotics, and they can cause damage to those tissues, so the tissues can't function as normal. Okay, so again, for the most part, most bacteria fall into those three, but there are exceptions. Okay, with bacteria, they will associate with other bacteria of the same species. They also associate with bacteria not of the same species, but what they'll do is that they will actually have an arrangement, a pattern in which they appear if you look at them underneath the microscope. So they can form clusters or they can form chains. So the chains are where we have two or more cells. If you only have two, usually we refer to that as like diplo, so diplococcus, or you could have tetrads that form. And so again, there are these arrangements that are formed that if you look under the microscope you can see. So in intrabio class you would have looked under the microscope and you would have seen staphylococcus being these clusters, or streptococcus being chains. Bacteria can be motile or non-motile, and so there are certain structures like a flagella that allows them to be motile. There's structures like fimbriae that allow for attachment, so the bacteria would be non-motile. So this is a figure that's from the food microbiology textbook, and so this just shows you some examples of some different bacteria. I included the other figure because I felt the other figure was a little clearer to see. I was trying to also fit this figure onto the slide, so I couldn't zoom in too much, but in that upper left panel, those are the rod-shaped bacteria. So there's little areas where they're strepto, so they're connecting end on end. And there's some that are also single. And then we have to the right of that in panel D, we have these chains are forming, and those are spears, so Streptococcus. And then panel E, which is back on the left side, we have these groups of 4 that are forming, so those would be tetrad. And then moving over to panel F, which is over to the right, we have these bacillus that have endospores. So again, if you kind of zoom in on that, you can see how they have like little holes in them, and that's where the spores or endospores are present. Okay. And then on panel G, which is going back on the left, the bottom panel, this is a Clostridium. So they have terminal endospores. So again, if you look closely, you can see these endospores. Their endospores are going to be terminal endospores, so they're found towards the end. Where in panel F, those are going to be central endospores, so they're found more in the middle. Okay, and then lastly, in panel H, which is the bottom right panel, this is where you can see they've done a special staining to show the flagella. And so you can see how we have flagella present, so those little string-like structures that are coming off of those bacteria. Again, as the legend indicates, this is Clostridium. So the Clostridium are rod-shaped or bacillus, and they can actually produce endospores, and they can actually produce toxins. And then they have flagella, so they're motile, and so Clostridium, species of Clostridium are medically relevant, and with foodborne illnesses they're relevant, and so definitely an important piece in food microbiology. And they're found in the soil, so it's relatively common that if produce is not washed and cleaned properly, that you could have Clostridium present. And again, because it produces toxins, because it can produce endospores, this becomes a huge issue. So looking at bacteria, when it grows on a nutrient agar plate, it forms colonies. And so in that colony, you would have thousands of cells present. Looking at the colony, you can look at different characteristics of it. So you can look at the edges or the margins, and you can look to see whether they're circular or they're irregular or lobulated. You can also look to see how the elevation, so whether domed or flat. Also the texture, so whether it has a rough or smooth surface, or whether it's shining or dull. And then we can also look at color, so whether it has pigment to it. And so all that information will help us identify the bacteria when it's growing on the nutrient agar plate. So there's information that we can find out when we're looking at it under the microscope. So that's microscopic observations, but there's also information that when it's growing on the agar plate without the microscope, we can help identify what it is. This is referred to as colony morphology, and the colony morphology is based off of macroscopic observations. So you can see in the figure to the left, you can see how these different colonies of bacteria have very different appearances. So all those pieces of the margin, the color, the elevation, the texture, the shape of it, again, can all can allow us to identify what it is. And again, back to that nomenclature, the name sometimes helps us to distinguish what it looks like or what it is. So looking at some of the structures that are found in bacteria, they're going to have cytoplasmic material that's going to be enclosed with a wall on the surface and then a membrane beneath that. Again, that's what the textbook's saying, but just note that not all bacteria have cell walls. So some bacteria lack cell walls, so they would only have a membrane present. And then the other piece is some bacteria also have an additional membrane, so they have the outer membrane. So back into intro bio class, you probably remember talking about gram-positive versus gram-negative bacteria. Gram-positive bacteria have a cell wall and a cell membrane. Gram-negative bacteria are going to have an outer membrane, a cell wall, and a cell membrane. Okay, and then again, we have bacteria that don't have a cell wall, that would just have a membrane. Okay, and those are usually your acid-fast positive bacteria, and again, they're the atypical. Most bacteria would fall into the category of either gram-negative or gram-positive. Okay. So for the bacteria, their cell membrane allows for nutrients to be transported from outside to inside or inside to outside. But the membrane also allows for energy generating components. So again, hopefully you remember this from intro bio class. So bacteria don't have mitochondria, they don't have organelles. And so they need to still make energy, they need to be able to make ATP, and they do that on their cell membrane. For their genetic material, they are going to have a bacterial chromosome, which is circular. They can also have plasmids that have small pieces of DNA, but again, both the bacterial chromosome and plasmids would be double-stranded, circular, okay, opposed to the linear chromosomes that we would see in eukaryotic organisms. This genetic material is not going to be enclosed, so it's actually moving, it's actually present, it just kind of aggregates together in the cytosol, okay? And there's no proteins that it's folding around, so there's no histones. So again, it's very kind of just crudely, it's like if you took an elastic band, right, that's circular, and you just kind of clumped it together and left it there, so that when it's needed, it's opened back up. They are able to transfer genes, so we do get genetic recombination, but they don't have gametes or zygotes, and they don't undergo sexual reproduction. So they actually will transfer genetic information through pili, which is a structure. Okay, it's like a straw-like structure that two bacteria can make connections to each other and then exchange these plasmids. And sometimes those plasmids have genes that code for things that would allow bacteria to survive, like antibiotic resistance or toxin production. Okay. For cell division, it's done by binary fusion. So, kind of mitosis, where you're going to copy the genetic information, you're going to have pinching in of the cell membrane, cell wall, and then the generation of two daughter cells. When we look at the ribosomes are going to be 70S, opposed to the 80S that we see in eukaryotic cells. So again, prokaryotic cells would have a 70S ribosomes, the bacteria do too. Their large subunit is 50S, and their small subunit is 30S. When they bind together, they just bind a little bit tighter, so we're going to get a 70S ribosome, and those are found throughout the cytoplasm. There is no endoplasm reticulum, and so there's no endoplasm reticulum to bind to. So the ribosomes will bind to RNA that's produced and make proteins right there in the cytoplasm. Prokaryotic cells, bacteria can also have flagella. Again, that allows for motility. They can have capsules and surface layer proteins. So they have receptors, they respond. So very much like, any kind of cell. Okay. And again, I've already mentioned when we were looking at the bacteria, that figure from the food microbiology textbook, where there's examples of endospores. But again, some bacteria are able to produce endospores, such as the Clostridiums, Bacillus, and Sporacina genuses. And all of those would be relevant to food microbiology because endospores, we would not want to pass those on into food products. So that's a consideration that needs to be taken into when you're processing food and then moving to retail, and then to the table. So as I mentioned in the intro bio class, you would have talked about the concepts of gram-positive versus gram-negative. On the left-hand side is our gram-positive bacteria, where it has a cell wall that has peptidoglycan as a subunit present in it, but it would also have surface proteins that are able to interact. And then it has the plasma membrane or the cell membrane present. So it's got these two layers. With a gram-negative bacteria, which we have on panel B on the right side, we have our plasma membrane present, our phospholipid layer. We also have our cell wall, but you'll see the cell wall is much thinner than our gram-positive bacteria. And then we have the outer membrane. Within the outer membrane, we would have transport proteins and surface proteins, but we also have lipid polysaccharide, or LPS. Lipid polysaccharide contains an endotoxin. Endotoxin means with a toxin within. So there is the endotoxin portion, but there's also a portion that's known as O antigen, which the immune system can recognize really well. And so with gram-negative bacteria, you can get the immune system to respond because of the O antigen, but then that causes the release of the toxin, and the toxin, it's known as lipid A, and so lipid A can get incorporated to any cell with lipids, which is any cell. And so if this is happening in, a host, a human, this means that this toxin, lipid A, can damage virtually any cell in your body. And so if there's high enough concentrations of it, it can actually cause multi-organ failure, septic shock, and death. So really important consideration that, if you have a gram-negative bacteria and there's extremely high amounts of it, that again is taken into consideration with food. And then our last microbe we'll be looking at is viruses. And so viruses are going to be a non-cellular organism. So they require a host cell. So they're known as obligate intercellular parasites. Parasites because they require a host cell. Also with the obligate there, intercellular, they go inside the host cell. So they're going to be composed of nucleic acid, either DNA or RNA, and some proteins. So again, this is a big distinction between prokaryotes and eukaryotic cells. They're going to have both DNA and RNA present inside them. With a virus, it's going to be one or the other. You are either a DNA virus or you're an RNA virus. Now, you could be a double-stranded DNA virus or a single-stranded DNA virus, or you could be a double-stranded RNA virus or a single-stranded RNA virus. But you're not DNA and RNA. You're one or the other. Okay, and that's how they're classified to begin with. So usually nowadays a lot of it's looking at their genomes and how closely they align, like making a phylogenetic tree. And so a lot of you have seen the phylogenetic trees of coronavirus with SARS versus COVID-19, and how do those relate phylogenetic. But originally how viruses were classified was based on which nucleic acid they had present, also based off of their size, their shape, in the shape of their capsid. The capsid is how these proteins arrange together to kind of form a head. Inside of that capsid would have the nucleic acid. So the job of the capsid is to protect the nucleic acid. So there are two basic types of viruses. There's bacterial viruses, so bacteriophage. So obviously those are very important because bacteria is very important in food microbiology. And so if we want a bacteria to produce food for us or to make a fermented food for us, and bacteriophage are infecting and killing them, that's going to affect the end product. But we can also utilize bacteriophages to actually help remove bacteria for us if we don't want bacteria present too. So the use of phages in food microbiology, I would say, is a newer area of research, just like it's a kind of a newer, not new, but like revamped way of thinking about medical microbiology and potential treatments. Instead of having to use antibiotics, could we use bacteriophage as a potential treatment if someone has an infection? Okay, so in this image, this figure is taken from OpenStax, their microbiology class. You can see this bacteriophage. It has these tail fibers. Those tail fibers would embed into the bacteria. And then there's the sheath. It's very much like a hypodermic needle, so that the nucleic acid, the viral genome, gets injected into the bacteria. That viral genome is being housed in the capsid. The genes in the viral genome code for the proteins that will make up the capsid, the sheath, and the tail fibers. So when the viral genome is injected into the bacteria is going to replicate the viral genome, express those proteins that the genes are coding for, so that new bacteriophages could be made. Now, because most bacteria have cell walls, the way these bacteriophages are going to be released is by lysing, bursting open the bacteria, and in that case, the bacteria dies, right? So a little bit different than when we look at like human viruses or mammal viruses. So looking at viruses that can infect humans or mammals, there are also viruses that can infect plants. Some of those viruses will have an envelope, and so if we look at panel A, which is on the left here, this is actually a naked virus or a non-enveloped virus. And so this virus has the nucleic acid. So for this virus, it's DNA. It has the capsomeres. The capsomeres are the proteins that make up the capsid. And you'll also notice that there's glycoproteins, these spikes that come off of it. Okay. Now, if you look on panel B to the right, this is an example of an enveloped virus. So it has the nucleic acid, for this one, RNA. It also has reverse transcriptase, so sometimes they bring enzymes with them, so we don't make reverse transcriptase. So the virus needs to make sure that it's bringing that enzyme to initiate the process. We then have our capsid that surrounds the nucleic acid. And then we have our envelope, and within that envelope, we have our glycoproteins. So with the viral envelope, that generally is coming from the host cell. So with an enveloped virus, when they exit the cell, they actually bud off. So it's like exocytosis, where like a vesicle is released, and so some of that host membrane actually gets incorporated and becomes the viral envelope. This gives it an advantage because the immune system can't see it quite as well, because it's your host, it's you that's making up this viral envelope. And again, we have these glycoprotein spikes that are present. So those glycoprotein spikes would be specific to that virus. Okay, so like an HIV virus would have different glycoprotein spikes than coronavirus. So coronavirus is an example of an enveloped virus, so it's going to have that envelope, but then it also has glycoproteins that allow for it to interact with ACE2 receptors. So the ACE2 receptors are found on cells in humans. So you have gut cells like tough cells that have ACE2 receptors, but you also have ACE2 receptors in cardiomyocytes and in your respiratory tract. And so you also have it in your mouth, on your tongue, are tough cells that allow you to taste and also in your nose. You have tough cells that allow you to smell. And so if you think about the symptoms with coronavirus, a lot of it has to do with cells that are infected and then get damaged and killed by COVID-19. Okay, just to kind of link this with some real world stuff. So during the semester, we'll be talking about some of the different viruses that are common for foodborne illness that need to be taken under consideration in handling of food products. The last piece is just to keep in mind that with viral capsids, those can vary in their shapes. So again, this is one of the ways in which viruses were first identified, because you could look at them under the microscope if you had an electron microscope, and you could look at the shape of the capsid. And we have a helical, which is in panel A, icosahedral. So icosahedral has 20 sides to it, and those sides come together to create this really geometric shape. And then we also have the miscellaneous other category. So sometimes these are called different things in different textbooks, but they're also, sometimes referred to as complex shapes. So again, there are these kind of 3 different shapes of those viral capsids, and then we can also have an envelope around those. So this is going to conclude the audio lecture that kind of does an overview of microbiology. Obviously, there's a whole lot more that you learned in 304 than just this, but some of those concepts and ideas obviously will be brought up during the course of this semester, but this is kind of the crash course, like the pieces, the morphology, and the structures you have to know for these different microbes. Thank you.